Hello friends, in previous lecture we talked about splicing mediated by Spliceosome, okay, and in that lecture we discuss it, and in this particular video, that is splicing mediated by Spliceosomes. This is part 2 introduction. I will give you a summary of what I have discussed in introduction part 1 of that is a discussion of the topic. Now, so I start with the summary. First point, here it is. You can see splicing of eukaryotic nuclear RNA precursor oblique heterogeneous RNA takes place in nucleus. Point number 2, spliceomal intron are largest class of intron found in mRNA primary transcript or hRNA in eukaryotic nucleus. Now, regarding spliceomal introns, so you know spliceosomal intron are intron whose removal is mediated by a large protein complex called spliceosome. So what is spliceosomes? It is a protein complex. Again, the point, very interesting point is spliceosome assembled during transcription process. That is, synthesis of RNA from DNA. And ATP is required for assembly of spliceosome. ATP is required. And the another interesting thing is that the process of splicing of a spliceomal intron by a spliceosome itself occur within a spliceosome. Okay, this is very interesting. That the whole process occurs within a spliceosome. Okay. Now the fourth point. In size, now we will talk about the size. Spliceosomes are as large as ribosomes, and their size is 3.5 megadalton to 4 megadalton. If you compare with ribosomes, well, when we talk ribosomes, the size of ribosomes is 2 megadalton to 3.2 megadalton. Well, this is size of ribosomes. This is size of ribosomes. So, size of both the this spliceosome and ribosomes are comparable, and obviously Dalton is unit for molecular weight, and it is equal to 1.66 into 10 to the power minus 27 kg, and m is obviously mega, that is 10 to the power 6. 10 to the power 6, you know it. So, due to its size, spliceosome is fit is large enough. It is large enough to fit nuclear pores. It is large enough to fit nuclear pores. That is, it does not pass nuclear pores. And due to this size and the corresponding property, due to the large size, spliceosome, what it does? It prevents who? mRNA precursor from what? From leaving or escaping the nucleus before mRNA precursor or precursors processing is complete. That is, the mRNA precursor processing is completed due to large size of spliceosome, which prevents the precursor mRNA to leave from the nucleus. So the point number 6 is, you see here, a spliceosome is a large multi-subunit complex made up of 5 RNA molecule whose total length is up to 100 to 150 nucleotides. Well, this number it may vary in different books. It may vary. So we are just putting also the word approximately, and 45 to 50 protein. So you can see you have RNA and protein, but you have only 5 RNA, small, and these 5 RNA molecules they have been named as a small n RNA, that is a small nuclear RNA. Okay, and again they have been designated as by these 5 letters, and the letters are U1, U2, U4, U5 and U6. That is, U3 is missing in the sequence. So you have to remember that U3 is not in the sequence. The sequence what you have for 5 RNA, 5 snRNA, a small RNA is U1, U2, U4 and U5 and U6. So obviously, as a student of chemistry or biochemistry, you know it, that symbol U is for uridine or uracil, and this nomenclature is based on the uridine content, and uridine is obviously the nucleoside of uracil, that is uracil plus ribose sugar combination you call uridine. And these U1, U2, U4, U5 and U6 snRNA are abundant. I think one N is missing. Abundant in eukaryotic nuclei. Okay. Now, since you talked that a spliceosome is made up of 5 RNA molecule and 45 to 50 proteins. So now, point number 8. A small nuclear RNA are always associated with a set of protein leading to formation of, and the resultant thing is called snRNP, that is a small nuclear ribonucleoprotein. Okay, and you also call it snRNPS, and this snRNPS is pronounced as SNRPs. So you see, you don't write SNR, you write as snRNP, but you pronounce it as SNRPs. Okay. Now, what I have said there that it will bind with a specific protein, but it has been found that snRNA in snRNP is similar to ribosomal RNA. Okay, it is similar to ribosomal RNA. Why? Because it directly incorporates both an enzymatic and a structural role. So in snRNP, the snRNA has both enzymatic and structural role. Now let us come to 9. Point number 9. You see here, SNRPs were, since it is a very important discovery, one of the important discovery when we talk splicing mediated by spliceosomes. So we, just as a glance for history, I want to tell you that it has been first discovered by Michael R. Lerner and Joan A. Stites. Later on, Thomas R. Seck and Sidney Altman also played very great role in their discovery and function and all this. And obviously these two gentlemen, scientists Thomas and Sidney Altman, were awarded Nobel Prize for Chemistry in 1989 for their work on SNRPs. Now again, what we have already told you that SNRPs are formed by the association of snRNA with proteins. So further detailing it, each snRNP contains 1 or 2 snRNA. So in one snRNP SNPs, you can have one, or you can have two RNA, okay, and a number of SNRNPs, a specific protein, and obviously they will be associated with a certain set of proteins, or which will be a specific for the two for the snRNA. Now further, it has been found that some of the protein which associates with snRNA, they can be common. They can be common to all snRNA. That is, some protein can be common to all snRNA. That is, they will get associated with the snRNA, and also others. So what about others? The others can be found. Sometimes they can be found only in one snRNA. Okay. So in the 10th point, you can see that SNRPs are building block of spliceosomes, and SNRPs we are repeatedly saying that it is formed due to binding of or association of snRNA with the specific proteins, one or two whatsoever. And SNRPs, so since we talked that spliceosomes formation, or rather it assembles during the process of transcription. That is, it does not pre-exist. So assembling takes place in a sequence. And what are the building block? Building block are SNRPs. So SNRPs assemble in sequence to form spliceosomes. So since the 5 we have talked U1, U2, U4, U5 and 6. So here it is U3, you can see the missing number. Apart from that, you have U8 and U13. These are also recognized as SNRNPs, which is found in nucleolus. You can see that it is found in nucleolus, but it does not take part. And are not part of spliceosomal complex. That is, these are not building block of spliceosome. So another is U7. This is also your SNRNPs. It is also not involved in splicing at all, but it exists. There is no any talk about spliceosomes, because it does not take place. Does not take part in the splicing at all. But it is responsible. It is responsible for what? For processing of 3' step loop of histone precursor mRNA. So it has a specific function. Now another two interesting SNRP is there, U11 and U12. These two are there, but their abundance in nucleus is low as compared to 5 SNRNA, that is U1, U2, U4, U5 and 6. In that respect, it is less. And what is its role? It is required. When it is required, U2 independent splicing process. So obviously you can, as the name suggests, there is a splicing process by spliceosomes in certain species in which U2, it is U2 independent. It is U2 independent. So now next, the point number 13 is there. Apart from SNRNPs, the another major another component of spliceosome is SR protein. Okay, so as you know, the S and R are a standard. These are a standard abbreviation of serine, amino acid, and arginine amino acid, and SR protein rich. Okay, rich rather we have to write rich SR protein content, rich SR protein content are involved in mRNA splicing process. Okay, so these help if it is there, component is there. Now, one specific thing about SNRs or SNRNPs is that these are major autoantigen. These are major autoantigen in spliceosomes. Okay, these are major autoantigen in a spliceosome, and as you know, when you talk at autoantigen, we are talking normal or complex protein that is recognized by immune system of patients suffering from autoimmune disease. Okay, so this is all about the summary of what we have discussed in previous my part 1 introduction discussion. Here is summary, and this written summary is there. It may help you. So thank you for watching. Please subscribe and share it my video to your friends and students. Thank you.